Hi there and welcome to Learning Made Easy. In today's video, we'll be discussing the major histocompatibility complex, MHC. MHC molecules are membrane-bound glycoproteins. Class I and Class II are closely related in structure and function. MHC molecules function as antigen-presenting molecules. Class III MHC molecules are unrelated to Class I and II, with different structure and function, but participate in immune response. Class I MHC molecules. Class I MHC molecules are composed of a 45 beta alpha chain and a 12 beta beta 2 microglobulin molecule. Alpha chain has three external domains: alpha 1, alpha 2, alpha 3, transmembrane and cytoplasmic anchor segments. Beta 2 microglobulin is similar in size and organization to alpha 3 and non-covalently bound to alpha chain. Alpha chain and beta 2 microglobulin have strong homology to constant regions in Ig. Alpha 1 and alpha 2 domains form a platform for peptide binding groove on top surface, large enough for 8 to 10 amino acid peptides. Alpha 3 domain and beta 2 microglobulin are organized into beta pleated sheets, known as the Ig fold. Alpha 3 domain is highly conserved among class I MHC and interacts strongly with CD8+ cell surface molecule. Class I alpha chain, beta 2 microglobulin and peptide are essential for proper folding and expression of MHC peptide complex on cell surface. Without beta 2 microglobulin, Class I MHC alpha chains not expressed on cell surface but can appear if transfected with functional gene. Class II MHC molecules. Class II MHC contain two different polypeptide chains, 33 beta alpha chain and 28 beta beta chain. These are membrane-bound glycoproteins. Each chain has two external domains, alpha 1 and alpha 2, beta 1 and beta 2. Membrane proximal alpha 2 and beta 2 domains are similar to immunoglobulin fold. Classified in the immunoglobulin superfamily. Alpha 1 and beta 1 domains form the peptide binding groove for processed antigen. Peptide binding groove of class II molecules is similar to class I. Comprised of 8 antiparallel beta strands and antiparallel alpha helices. Peptides can bind in range of 13 to 18 amino acids. Lacks conserved residues in class I that bind short peptides. Forms an open pocket compared to class I's socket-like opening. Class I and II MHC molecules have several hundred allelic variants, but each individual expresses only a limited number. Peptide binding by class I and II MHC molecules is promiscuous and not specific like antigen binding by antibodies and T-cell receptors. Both class I and II MHC molecules bind peptides in an extended conformation, with class I binding peptides of 8 to 10 amino acids and class II binding peptides of 13 to 18 amino acids. Class I binding requires specific amino acid residues near the N and C termini, while class II binding has no such requirement. Peptide MHC association is stable with Kd values ranging from 10 to the power of -6 to 10 to the power of -10. Most of the expressed MHC molecules are associated with a peptide under physiological conditions. Class I MHC molecules bind peptides and present them to CD8+ T cells. The peptides are derived from intracellular proteins and are processed through the cytosolic or endogenous pathway. Each nucleated cell expresses several unique class I MHC molecule, with its own rule for peptide binding. Peptides expressed on surface of cell vary due to inheritance of different MHC class I alleles. Bound peptides are 8 to 10 amino acids in length, with most being 9, and contain specific amino acid residues at key position. Class I MHC to bind to diverse peptides is due to presence of same or similar amino acid residues at key positions. Anchor residues in peptide are complementary to binding groove of the class I MHC. Chemical identity of anchor residues is determined by the varying amino acid residues lining the binding sites in different class I allelic variants. More than 2,000 distinct peptides were found in a study of peptide binding by class I MHC molecules. All peptides that bind to class I MHC contain C-terminal anchor, mostly hydrophobic residues. Knowledge of key positions and chemical restrictions for amino acids may allow for more targeted clinical studies, such as vaccines. X-ray crystallographic analyses show that peptide binding groove in a class I MHC molecule can interact with a broad spectrum of peptides. Anchor residues at both ends of the peptide are buried within the binding groove, with main contacts involving residues 2 and 9. Peptides arches away from the floor of the groove in the middle, allowing for accommodation of slightly longer/shorter peptides. Class II MHC molecules bind and present peptides to CD4 plus T cells. Peptides associated with Class II MHC are derived from self or foreign proteins processed through the exogenous pathway. Peptides in Class II MHC peptide complexes contain 13 to 18 amino acids, longer than those in Class I molecules. The peptide binding groove in Class II molecules is open at both ends, allowing longer peptides to extend beyond. A central core of 13 amino acids determines peptide binding to Class II. Peptides that bind to Class II MHC often have internal conserved sequence motifs. Hydrogen bonds between the peptide and Class II molecule are distributed throughout the binding site. Peptides that bind to Class II MHC contain an internal sequence of 7 to 10 amino acids with hydrophobic residues. Over 30% of peptides eluted from Class II MHC contain proline residues at positions 2 and C-terminal end. The relative flexibility of prolines contributes to peptide binding promiscuity in Class II MHC. MHC is a collection of genes on chromosome 6 in humans, human leukocyte antigen, LA, and chromosome 17 in mice, H2. The MHC genes are organized into regions encoding 3 classes of molecules. Class I MHC genes encode glycoproteins expressed on nearly all nucleated cells and present endogenous peptides to CD8 plus T cells. Class II MHC genes encode glycoproteins expressed on UPs, macrophages, dendritic cells, and B cells, and present exogenous antigenic peptides to CD4 T cells. Class III MHC genes encode various proteins with immune functions, including components of the complement system and molecules involved in inflammation. Class I MHC molecules are encoded by K, D, and L regions in mice, and by A, B, and C loci in humans. They consist of an alpha chain and a common beta-2 microglobulin chain. Class II MHC molecules are encoded by Ya and E regions in mice and by DP, DQ, and Dr regions in humans. They are composed of alpha chain and beta chain, and multiple genes can be present for either or both chains. Some additional genes in Class II region encode non-classical Class II molecule with specialized func. Class III MHC products include complement components and inflammatory cytokines and have little in common with Class I or II molecules. MHC genes are highly polymorphic with many alternative forms or alleles within the population. MHC genes are closely linked, meaning they are inherited as a set or haplotype. Humans are heterozygous MHC loci, meaning they have different alleles contributed by each parent. Inbred mice express identical MHC molecules. They have identical maternal and paternal haplotypes. MHC molecules exhibit codominant expression. Both maternal and paternal gene products are expressed at the same time and in the same cells. When two mice with different MHC haplotypes mate, the F1 generation inherits both parental haplotypes and is histocompatible with both. The parental strains can't accept a graft from its F1 offspring due to MHC molecules from the other parent being viewed as non-self. In humans, each individual is heterozygous at each locus and the MHC genes are usually inherited as a haplotype. It's rare for two unrelated individuals to have identical genes. Transplantation between non-identical individuals is challenging. Clinicians look for partially histocompatible family members or rely on donor databases for MHC match. Even with partial matches, heavy doses of immunosuppressive drugs are needed to inhibit rejection response. MHC region has multiple genetic loci encoding proteins with same function, leading to polygenic region.
LA, B, C can present peptides to CD8 plus T cells and LA-DP, DQ, doctor to CD4 plus T cells.
Expression of multiple MHC alleles at each locus leads to diversity in individual expression. MHC class II have more potential for diversity. It has two different polypeptide chains from different loci. Heterozygous individuals can express combinations from same or different chromosomes. Humans have three classical class II genes, DP, DQ, doctor, leading to expression of 6 identical and 6 new alpha, beta combinations. Diversity of peptides displayed by MHC molecules increases the number of antigenic peptides presented, beneficial in fighting infection. Generation of T and B cell receptors is dynamic, but MHC molecules expressed by individual are fixed. The host's ability to respond to unexpected environmental changes is due to its flexibility in MHC. MHC is one of the most polymorphic genetic complexes, has extraordinary no. of different alleles at each locus. The no. of alleles in human La class I genes is approximately 2013a, 2605b, and 1551c alleles. The polymorphism in the human MHC is enormous, with over 1.7 billion class I haplotypes and over 1.7 times 10 to the power of 24 combinations of class I and II alleles possible in the entire human population. A reduction in MHC polymorphism can predispose to infectious diseases. High levels of MHC polymorphism provide survival advantage. MHC diversity may protect from wide range of infectious diseases. So, there you have it, a brief overview of the 3 classes of MHC molecules and their functions in the immune system. Thanks for watching and be sure to subscribe for more learning made easy.